from Pennant Hills High School. She will be running you through non-infectious disease and disorders and looking at some exam approaches for this module. And I know Halima would love to hear some questions from you. So if you have any questions or anything to discuss, please, again, throw it in the chat and I'm sure she'll be happy to answer. Over to you, Halima. Thank you.
Well, good afternoon, everyone. I hope you've had a chance to have some lunch, and I'm sure your brains are really full from everything you've learnt today. But we need to take a big breath and get ready for this last lap. Now, by way of introduction, I am a biology teacher at Pennant Hills High School, but I also have another aspect to my job, which is curriculum writing specialist. So for the past 18 months, I've been working with the department to look at how we can teach writing better as teachers to students. So I don't know what your experience has been in high school, but I know mine was certainly, they'd say, now, Halima, we want you to write better. We want, more depth. And I'd say, well, how do you do that? And they go, well, kind of, you just do. And I thought, well, gee, that's not helpful. So what I'm going to do today is through the content, we're actually going to have a look at how we can break down some questions and some of the, I guess, parts that go into that to make it a little bit easier for you. Now, I also just wanted to mention, I'm not sure exactly where everyone's up to in Module 8 or even if you've started it. So if you're happy to just throw into the chat where approximately you are in Module 8, that would be really helpful for me. But essentially, the way I'm going to run it is we're going to do a few activities and data processing questions that are all about non-infectious disease, but also you don't need to know a whole heap to participate in them. So I'm hoping it will give you a bit of a background. We won't cover absolutely everything, but I'll do as much as I can in the time. So let's take it away. Now, you can see the scope here for non-infectious diseases. There's basically the 4 parts. We've got homeostasis, causes and effects, epidemiology, and technologies and disorders. So I would say to you, when you're studying, it's really important to go back to your inquiry questions and think, can I give a really holistic answer to these inquiry questions by looking at all the information that I've learned about the dot points? Because each of those inquiry questions could, in fact, be turned into an HSC question. Now, I think part of this you'll be really familiar with, that in order to answer an HSC question, we really need to understand that question, we need to plan for it, we need to use a whole lot of subject-specific terminology, so all that vocabulary that your teachers are always saying that you must know. But I think there's also some little hidden parts, so you really have to write with an appropriate text type. You need very succinct writing, logical communication, and cohesive writing. So we're going to touch on a few of those things as we go through. Now, those of you who do maths, and all of you would have done it at some point, you know that there are secrets to algebra. So 3x means 3 multiplied by x. And so quite often when students come along, if they don't know that little secret, they really think that algebra is something that they can't do and is really hard. But as soon as they know these little secrets, it makes it a whole lot easier. And I think writing is very much the same. So what we can see here is we've got a marking rubric where it's saying provides a thorough explanation of how educational programs reduce the incidence of non-infectious disease and supports the answer with suitable examples. So if you wanted to get 5 out of 5, you would need to do that. But my question is, well, what exactly is thorough? So let's have a little bit of a look. Now, these particular examples that I've stolen from the New South Wales Education Standards Authority, what we've got here is an answer that's on the cusp of band 4 to 5 and also an answer that's on the cusp of band 5 and 6. And what you can see from the annotations, and this is all provided, this is all up there to see, if we're looking at the band 4 to 5, they're saying the response communicates effectively the impact of education programs on the incidence of skin cancer using correct scientific terms. Now, if we pop up to the band 5 to 6, what we can see here is the response communicates scientific understanding succinctly and uses correct scientific terms. So remember, this is not yet a band 6, it's just on that cusp of. So let's go a little bit further into some of those terms. So those terms come directly from the performance descriptors, which comes directly from the biology syllabus. So in order to be answering at a band 6 level, what they're saying is that you need to communicate scientific understanding succinctly, logically, and consistently using correct and precise scientific terms and application of nomenclature in a variety of formats and a wide range of contexts. Now, if you jump down to band 5, the difference here, you can see it has to be logical and effective, and it still has to have correct scientific terms. but they're also, the one that's missing is succinctly and consistently. So all the time. So let's jump in and have a little bit of a look at what some of these words actually mean. So if you're being succinct, clear and compact, expressing what needs to be said without unnecessary words. So in my experience, some students who are really good at English can struggle a little bit with bio because one of the tricks with bio is we're only given a very limited space, and they're asking for us to be able to put a heap of information into a really small space. And being succinct is how we do it. So we don't have unnecessary words. We don't just reiterate the question, because if you rewrite the question but don't add any new information, that doesn't get you any marks. Now, of course, we need to logically sequence our answer so that it helps the reader to understand what it is we're trying to communicate consistently all the time, and of course using correct and precise scientific terms. So it's not just about throwing the words in, it's about using the best word in that situation for your communication. And of course, nomenclature is just naming systems. So for bio, we'd want something like E. coli to be written correctly. And of course, a variety of formats. They might ask you to write a procedure, an explanation, a discussion. They might ask for part of a report, like a method. So the trick here is, I always teach my students, if you're writing a method, then you would write it as a numbered sequence and you'd start with an action verb. So we're telling the reader what to do. Line 3 Petri dishes with cotton wool, place 3 identical seeds into each Petri dish, and add 5 mils of water to each dish. So if you want to answer really well, we need to understand, communicate using suitable language and for a specific audience and purpose. So we're really trying to show them what we know, and we have to do this under time pressure. So, no problem, we can do this. So what I'm going to do is I'm going to get you to do a series of little activities as we go through. So I'm hopeful that you've got some workbooks printed out, or if you don't, you have some access to this, or if you've got some paper. So for this first one, in terms of understanding the actual question, one of the techniques I like to teach my students is to circle the verb, underline the content, because you really want to get your mind and say, which part of the syllabus is this coming from, and what did my teacher teach me about this? And also place a box around the limiting words, because one of the things with biology is you could almost write forever because there is so much to know. But for an HSC question, we want to know what is the specific thing that this examiner is looking for and no more. So to that end, what you have in front of you, hopefully on about page 5, I'm guessing, or 6, let me see. Yep, page 5 is explain how educational programs can be effective in reducing the incidence of non-infectious diseases. Support your answer with examples. So if I can get you guys to grab your pen or pencil and see if you can put a circle, an underline, and a box, and I'll give you about 30 seconds to try that one. Okay, let's jump in and have a look and see how you went. So hopefully you should have a circle around explain how. We should have a line underneath educational programs can be effective, because we're looking at that efficacy. And also we've got a box around non-infectious diseases, because they're not asking us about educational programs to do with infectious diseases, just non-infectious diseases. Now, the next thing we want to do is we want to plan our answer. Now, obviously you won't have time to set something like this up in the HSC, but if you've done this in your practice, it's a really good way of ordering in your mind how you're going to tackle it. So the first thing we want to know is, do we know what an educational program is? So public health programs use education to change the behaviour of members of the population to reduce their exposure to risk factors that can cause preventable diseases. We need to make sure we understand the all-important incidence, which of course is the number of new cases of a disease within a specific time period. And of course, non-infectious diseases are those that are not caused by a pathogen or are not contagious. Then, of course, we need examples. So you need to think of 2 examples. Now, I don't want you to worry as I'm presenting if any of the examples are unfamiliar to you, because a lot of teachers use a really wide variety of examples. And if these are not the ones you've used, that's okay. It'll give you an extra string to your bow, or you can use the ones that you know. So, of course, the Slip Slop Slap campaign, this was right when I was a little kid, and they've added a few extras to it. And of course, the Quit campaign, which with all that plain packaging or the graphic images. Now, because this is an explain question, the other thing we really want to think about is that we need to be using words such as resulting in, leading to, because of, due to, therefore, causing. These are all the kind of cause and effect explain type words. So when we jump onto the next page, what we're going to see here is I've written you, or I've found you an answer, but what we want to do is just pull it apart a little bit and have a look. So as you're looking at this, we're going to read through it and we're going to see if you can recognize as I'm reading some of those science words that are in green. We're going to add some pronouns in yellow, examples in pink, and explanatory language in blue. So we'll read through it first and then we'll have a look. So public education programs are based on data from epidemiological studies linking causative agents to non-infectious diseases. They use education to promote behavioural change that will lead to a reduced number of new cases of preventable diseases, and in brackets there, incidents. Education is provided via advertising such as posters, radio, television, and social media. The link between high energy UV light exposure and the development of melanoma led to the development of Slip Slop Slap program. This program educated the public about the dangers of UV exposure and encouraged behavioural changes to reduce exposure, such as wearing a shirt, hat, sunscreen, sunglasses, and seeking shade. This has led to a decrease in the incidence of skin cancer in the under 40s. Tobacco is the leading cause of preventable death in Australia, with 2/3 of smokers dying of a smoking-related disease such as cancer. The Quit campaign and anti-smoking education program saw the introduction of plain packaging, graphic images of cancer, and the introduction of a national Quit helpline. While these measures resulted in fewer smokers, a decrease in the incidence of smoking-related diseases should be seen in the future. Now, as we click through this, all of those green ones, and I don't think I've even got all of them, but you can see there's a whole stack of that subject-specific vocabulary, or in other words, our science words. Now, when we click again, what we'll see here is we've got in pink some examples. We've got some pronouns in yellow. Now, just to remind you, pronouns stand in the place of a noun. And if we have a look at this first one, they use education to promote. We have to think about what is they? They is a public education program. When we come down further, we can see this program educated the public. Now, which is this program? Slip Slop Slap. And by using these pronouns, it allows us to essentially refer back to something that is a long name with a very short word, which again helps to make us succinct. And of course, our blue, we've got a whole lot of explanatory language. So we're using education to promote. That will lead, such as, lead to the development of. So if you have written an answer and you go through and actually annotate it like this, you will see what you've done really well. So it can be a very helpful technique. Now, moving on to homeostasis. Now, homeostasis is how we regulate an organism's internal environment and we maintain it despite any external changes. So moving on from there, we're going to have a little look at describing one mechanism by which plants maintain internal water homeostasis. So again, we're going to jump onto page 8. Now, if you're good and you don't have a look at the answer, you can give it a go yourself and say, where do I put my circle for my verb? What do I underline for my content? And let's place a box around any limiting words. So I'll just give you 10 seconds to do that. And of course, what we have is describe. So we want features and benefits essentially, but we're describing one mechanism. So if we're describing a mechanism, we're describing how, and we're talking about plants maintaining internal water homeostasis. So let's have a look at how we would plan an answer for that. So first of all, we need to know that a mechanism is a process. We need to understand that homeostasis is maintaining that stable internal environment despite variations in the external environment. And of course, you need to think of a mechanism. So here we're looking at, we've got guard cells controlling pore size of a stomate. And of course, some of the science words that we're going to want to be using are things like guard cell, stomate, gases, water, vapor, plant hormones, abscisic acid, etc. And of course, we're using descriptive language. So we're really trying to choose words that are going to help the reader understand that process of stomates opening and stomates closing to regulate water loss. So again, what I've got here is an answer for you, and we're going to have a little bit of a look at this one. This is one of the HSC answers that has an annotation. So let's read through it. Closure of stomates during the hottest part of the day is a mechanism used by plants to prevent water loss via transpiration. And hopefully we all remember that transpiration is that movement of water from the roots through the xylem all the way up and out through the leaves, thus helping maintain internal water homeostasis. When stomata close during the middle of the day, guard cells become flattened, thus restricting the transpiration of water. And of course, I would use the word flaccid rather than flattened. The pig-faced plant is an example of a plant which closes its stomates during the hottest part of the day to conserve internal water. This aids in homeostasis. So when we jump over and have a look at the annotation, we can see that the marker has said that the response has used correct and precise scientific terms. They've applied nomenclature to describe the structures, so they've used the right words. They've logically communicated the linking of a guard cell closure and reduced transpiration, but the response did not address the mechanism by which the cells become flaccid. So this is on the cusp of a band 5-6 again. So if we have a look, it's got a bunch of those sciencey words in it. And of course, that link back to the question. Now, for activity 3, I'm going to get you to jump onto page 9. So we're going to read through the answer in your booklet, which is actually on page 10, just tricked you. So I'll just pop that in the chat so we can all be in the right place. Page 10. And I'm going to get you to have a read and see if you can highlight or circle or underline some of the subject-specific terminology and highlight any word groups that describe that mechanism for maintaining internal water levels. So I'll just give you a couple of minutes to do that, and then I'll show you an answer. And I'll just check the chat in case anyone's got any questions or comments. Okay, let's see how you've gone with that one. So as I click through, you'll see a bunch of subject-specific vocab. So these tend to be words that we're just seeing in science. We're not using them when we're answering our food tech or probably not chemistry either. You can see here we've got our descriptive word groups. So are made up of and is regulated by, regulate the size, causing the water to follow. And of course, we've got a couple of pronouns there in yellow. Sorry, that went quickly. Let me just move back to that one for you. So compare your answer and see what you found and see what you'd like to circle. Now, the next few pages, you can see I've got up here, what I've tried to give you is some background information. There probably isn't time to go through all of it today, but just looking at this next section, it's really important to have a good understanding of the nervous system and the fact that we've got the central nervous system, which is the brain and the spinal cord, the peripheral nervous system, which is all the rest of your nerves. I remember I've actually just taught my year 9s at the end of last term what a nerve looks like. And we've got the dendrite, which is that large spiky bit, and then the axon where the nerve impulse travels down, and the axon terminals at the end. So there's a bunch of stuff in there that might be helpful to you. And so we'll just skip over those for the moment. And we will jump on to we'll have a look at thermoregulation. So essentially, when we're looking at this section of the syllabus, we're thinking about homeostasis and maintaining that stable body state. Now, if we look at something like thermoregulation, we know that when we've got a negative feedback system, it's always working to counteract the change. And so what I mean by that is when you have proper temperature and you get a little bit cold, your body is going to work really hard to heat you back up to your stable state, which, as we know, is somewhere around 37 degrees. And of course, if you get too hot, your body will work very hard to cool you down. Now, the way that it can do that, there's a few different things. We've got blood vessels which can vasodilate, which means that they essentially just open up and they're a little bit closer to your skin. That's why our cheeks go red when we get hot, because we're trying to get rid of that heat. We've got sweat glands that can sweat. And of course, in this day and age of COVID, we can't do it anymore, but I usually get my students to lick their hand and blow on it, which then you can feel the cooling, but we don't want to spread the COVID around. And of course, our cells can decrease their metabolic rate to decrease heat production, because you tend to feel a little bit lethargic in the heat. And of course, when we go over to the body too cold side of things, of course, our blood vessels can vasoconstrict, which is why we often end up with very sort of pale fingers. We can shiver to create heat, and our cells can increase their metabolic rate to generate heat. And of course, our hair erector cells can cause us to get goosebumps to try and lift up our not very well insulating hair, since humans don't have a heap of hair. Now, again, what we need to think about is the fact that we have a stimulus, which is the temperature. That stimulus is detected by thermoregulators either in the skin or our hypothalamus, and then that message is sent through our central nervous system, so through our interneurons, and then is passed on to the motor neurons when our control center, which is our brain, has decided what to do. And then it tells those effectors, or maybe it's the sweat glands or the muscles, that they need to do something to actually change the situation. So I've got a little HSC question for you on page 19. Now, this shouldn't take you long, and if you need any help, I'll leave this one up so you can see. We just need to fill in those 3 boxes on page 19. So I'll just give you a moment to do that. Okay, let's have a little look and see if you put something like this. So change detected by thermoreceptors in the hypothalamus. Then the nerve impulse travels to blood vessels and causes vasodilation. And of course, on the other side here, the nerve impulse travels to sweat glands. Sweat's produced and causes evaporative cooling. So, oops, we'll just move back to that one for a second. So if you manage to get those three, you just got 3 marks in your HSC. So fantastic work. And we know we tend to say that it's 1.8 minutes per mark, but you did that even faster than that. So look at you go. Now, moving on to look at some glucose regulation. So glucose regulation is interesting in that we say that it's predominantly controlled by the endocrine system as opposed to the nervous system, which, so if they're asking you for an example of homeostasis with the nervous system, we talk about temperature, and for the endocrine system, we tend to talk about temperature regulation. Now, sorry, endocrine system, we talk about glucose regulation. Now, when we have a look, this is a little bit of revision from junior school. Remember that when we eat, we digest our food and then it goes through our digestive system. And at what point does the sugar get sucked out? I always do this, and I'm hopeful that when you see this, you're reminded of the villi in the small intestine, which increase surface area. So we remember that the villi are very closely associated with a whole lot of blood vessels, and so that glucose can then enter your blood. And as you know, you eat a chocolate bar and then you think pretty soon after that, oh, I've got a nice sugar high. So as that glucose goes into your bloodstream, because our bloodstream contains or should contain a very set amount of glucose, whenever we disrupt that, a negative feedback system kicks in and we have to, it has to get it back to that stable state. So when we have a look at what's happening here, we've got glucose in our bloodstream. And of course, we then have to have something to detect that. And that something is the pancreas. So you can see here on the slide, we've got the endocrine system with all of its glands, and the pancreas, which my students always tell me looks like corn, produces insulin. Now that insulin, being a hormone, will go around the bloodstream. And as it goes around the bloodstream, it will pass certain cells like muscle cells, like liver cells, and it will stick onto the receptors on the top of that cell. Now, when it does, it tells the cell that it wants it to take in that glucose because we want to get the glucose out of the blood and into the cell. Now, remembering back to year 11, cell membranes are very selective. They don't let just anything come in and out. It's like the bouncer at the nightclub who says, no way, you have ugly shoes, you cannot come in. So we want that glucose going in and we can store it as glycogen, and that brings our blood glucose down. And of course, on the flip side, when you haven't eaten for a while and you're sitting in class and your stomach's rumbling a bit, what will happen is your pancreas again will detect this and will actually pump out some glucagon, which will cause your cells then to release some of that glucose. Now, I've got a great HSC question here for you. Now, this one is based on this graph. So let's just have a little bit of a look at the question. Use the data provided to explain how blood glucose is controlled in the body. So they want your understanding, but they want it based on this particular set of data. So what we can do here is we can, again, have a look at what are we going to circle, what are we going to underline, and what is the limit here. So we're going to explain, and it's really explain how. We're looking at blood glucose control, and we're looking at using the data provided. So let's look at some definitions. Blood glucose is just the amount of glucose in the blood. Homeostasis is maintained by the hormones insulin and glucagon. And of course, let's have a little look at what we're seeing in the graphs, because that's really important. So you've got a picture on page 20 of your graph. So if you can have a look at that while we read out the description. So you can see that glucose increases really quickly for 1 hour after the meal and then decreases to almost pre-meal levels, which is about 4 to 6 millimoles per litre between hours 2 and 5. And what I would suggest for a question like this is once you have read the actual question, it's really important to go in and annotate your graph and pull out the important things, because then you can refer back to that in your answer. Now, secondly, we've got insulin. So the insulin increases for an hour after the meal and then decreases between hours 2 to 5. And of course, the glucagon, which increased before the meal because you were hungry and you didn't have enough glucose. And then it decreases after the meal for the first 1.5 hours, but then gradually increases, but not quite to pre-meal levels. And of course, we've got our explanatory language at the bottom. So we're going to have a little bit of a look. Now, this answer actually goes over 2 pages. And before we go any further, there's one little mistake that I want you to correct, which I thought I had corrected out, but I missed it. Now, what you'll see here is about just after halfway down, you'll see it says, to counteract this change, release of insulin by alpha cells. Now, who can tell me the problem with that statement? Anyone who's learnt about it? Which cells release insulin? It's actually the beta cells. So if you could cross out alpha and write beta for me, that way your teacher will know if you're really listening or I'm boring you to death. Fantastic. So let's have a look at this answer. Homeostasis of glucose levels in the body is maintained via a negative feedback loop controlled by the hormones insulin and glycogen. Should be glucagon. This is a terrible one. The digestive system breaks down food consumed to release glucose and other components, which is represented by carbohydrate meal at hour 1. Release of glucose from the meal can be seen by figure 1 by the rapid increase in glucose levels within the first hour from 4.25 to 7.75 millimoles. So what I want to draw your attention to here is that we're actually referring back and using specific data from those graphs to show that we understand, because I think this question is not only do you understand the regulation of glucose, it's also about do you understand the graph and can you read what's going on there. So to counteract the change, release of insulin by beta cells, we've changed that one, as seen in figure 2, corresponds with the rise in glucose, causing cells to take up glucose and the liver to convert glucose to glycogen for storage, thereby decreasing the blood glucose. This can be seen in figure 1 with the glucose levels decreasing from the peak before hour 2 to hour 5. Now, this continues on. So because it's a 6 marker, I want you to really think about the fact that you've got to give a really holistic understanding and you want to give the marker enough to give you 6 marks. So if they've given you this amount of space, do try and fill that space, but not by repeating yourself. So prior to the meal, high levels of glucagon were causing glycogen stored in the liver to be converted to glucose and released into the bloodstream in order to maintain optimum glucose levels in the absence of nutrients being consumed. See figure 3. So that's important that we're referring back to the graph. This same graph also illustrates that the moment the meal is consumed, causing glucose levels to increase, the glucagon levels rapidly increase in the first hour, after which they slowly increase to approximately 100 micrograms per milliliter, still well below the peak experienced prior to the meal. This data is to be expected as the beta cells producing glucagon. Now, that's the other one we have to change because we know that it's the alpha cells that produce glucagon. So if you could just cross out beta and write alpha for me, that would be fantastic. Detect a rise in glucose and hence reduce production. The opposing actions of the hormones insulin and glucagon work together in negative feedback loops to maintain a steady state or homeostasis of glucose levels to maintain important functions such as cellular respiration that are vital for survival. So that was a really big one, but did we answer the question? Use the data to explain how blood glucose is controlled in the body? I think yes. So if we have a look here, I've annotated this one for you so we can see all of those yellows, greens and pinks. And what you will see is there's not a lot that isn't highlighted, which really shows that it's important to make sure that all of your words are or add meaning. If you've written something down and it doesn't add something extra, think, is it worth writing down? Okay, now we're going to move on to temperature regulation questions. Now, this one is outlining humans, how maintenance is different to the way that glucose is controlled. So the interesting part with this one is whenever I'm doing similarities or differences, so it's really compare, I want to look at the same thing for each system. And what I mean by that is I always draw a quick table so I can compare similar parts. So the main messaging system involved for the nervous system, for temperature regulation is the nervous system, whereas glucose regulation is the endocrine system, insulin and glucagon. Now, if we're looking at receptors, so temperature is thermoreceptors in the hypothalamus, whereas glucose is cells of the pancreas, because the idea is if you can make it really clear and really easy for the marker to give you marks, they will thank you for that. Of course, when we're looking at our effectors or the things that actually cause something to happen, for temperature, we've got muscles that shiver, whereas on the other glucose side, we've got cells that break down glycogen in response to increased concentration of glucagon. And of course, the response is temperature increases or blood glucose increases. So having a table like that then very easily allows you to go in and answer the question. So I've got another little job for you here. Let's have a look at the sample answer and see what mark you would give this sample answer. So we'll read it together. Temperature changes are detected by the hypothalamus in the brain, but changes in glucose are detected by the pancreas. The response to changes in temperature is via the nervous system, but glucose is regulated via hormones. Now, when we jump up to the marking guideline, we can see outlines differences between control of blood glucose and body temperature. And of course, when it says outlines differences, that implies that there must be more than one. And have they done that? Yeah, I think they've done that. I think I'd probably give this a 3. I mean, I would have fluffed it out a bit more, but I think it sort of does the job. Now, this one here, when we have a look, has got a little bit more, a little bit more in there. Say that one's a little bit blurry, so we'll move on from that one. Now, this one, glucose is a chemical that must be maintained at concentrations between 70 to 130 milligrams per deciliter in the blood in order for the body to function normally. They're asking you to draw a diagram that illustrates how the body maintains blood glucose within this range. So what are we going to draw? I'm going to show you, because this one takes a little while. I always tend to go for a figure 8, and the place where I start is stable state. So we've got what we want in the middle, and then we have to say we've got the stimulus. So what's going to happen if it gets high and what's going to happen if it gets low? And I've annotated that around. So I think it's important, if you haven't drawn a diagram like that, to do that, because it's something that could pop up in your HSC. Now, this one I'm going to get you to give a go. So we're now on page 25, because this is a really quick one that just requires you to draw a couple of lines. So let's have a look here. Sketch a graph on the axis provided showing the expected blood glucose levels of both a healthy person and a diabetic person after consuming fruit juice. On the same graph, show what would happen when the diabetic person injects themselves with insulin 20 minutes after consuming fruit juice. So just remember, for those of you who may not have studied this yet, if you're a diabetic, let's just go with you've got the type of diabetes where you can't produce insulin. Think about what's going to happen to those sugar levels if you can't produce insulin. So I'll just give you a minute to see if you can draw a couple of annotations onto that set of axes.
There's a question there, Halima, about going back slide 36 for 10 seconds.
Absolutely. Now, slide 36, there we go. Okay, let's dive in and have a look at how you went. So what we're looking at here is we've got blood sugar level up here, because remember that's the thing that we're measuring, and we always put our dependent variable on this axis. And then we've got a couple of annotations down the bottom because we're looking at the time after drinking juice. Now they've put juice here. I'd probably be inclined to put it there, but that's okay. And essentially we can see we also need to have a key. So we've got the diabetic person, and now they've said the normal person. I'd probably say non-diabetic person, because I think you could be non-diabetic and not particularly normal. So we've got a big spike here in sugar because we've had juice, and juice is very sugary, and that's then going to slowly come down. Whereas the person who can produce insulin, we're going to have a bit of a spike. Our pancreas is going to say, hold on, here's some insulin, and bring that right back down to stable state. So if you got anything like that, fantastic. If you didn't, it's okay, because now you know, and if you see something like that, you'll be able to answer it. Now we're just going to skip on ahead to causes and effects. So I think I've given you a bit of information in your booklet on page 26. And at this point, I also just wanted to show you a really good little resource if you haven't come across it already. So if you dive in and Google NESA biology, and then scroll all the way down to assessment and examination materials, what you'll see is additional sample HSC examination questions. These are really useful for you to practice because, as you saw on the very beginning of my presentation, it doesn't matter how many multiple choice questions you do, the place where you really add value to what you're going to do in the HSC is answering those extended responses. So it's really important to do as many of those as you can. Now, we're going to get stuck in and do a few more activities here. So we're skipping past page 28, which has got more information on it, and we're diving onto page 29, which is all about mesothelioma. So what we're going to see here is mesothelioma is a disease that occurs when asbestos fibres are inhaled and become embedded in the protective lining of our lungs, which is called the pleura. Now, the only known risk factor for mesothelioma is exposure to asbestos, and it can take many years after being exposed for mesothelioma to develop. So a lot of fibro houses have asbestos in them, so it's no problem living there. But if you were going to actually knock it down, then these days there are really strict rules in place to make sure that when that happens, nobody gets affected. So the kind of people who get exposed to asbestos at work can be builders and plumbers, electricians, welders, asbestos miners, of course, and automotive industry workers. And I suppose if you're wondering what's so good about asbestos, it's really fire resistant, which is why people tended to use it. Now, unfortunately, Australia has one of the highest incidence rates of mesothelioma around the world, and this rare and aggressive cancer has no cure. So between about 700 and 800 people are diagnosed each year, and the average time between diagnosis and death is around 11 months. So it's really not something that you want. So what I've got is some data here, and our question is describe the trend for people in the graph above. Now, I would want to mention whenever you get a describe the trend, it's really important to mention both of your variables. So you want to mention number of cases and you want to mention time. So I'll just give you about 30 seconds to think about what you might write, and then we'll have a look at an answer. Now, for anyone who wanted to answer it increases, you wouldn't get the marks because that is an identifier and doesn't really show all of your knowledge. So we're describing, so we're going to say the number of new cases of mesothelioma in Australia increases over time from approximately 150 cases in 1982 to a peak of 775 cases in 2013. After this time, the number of cases starts to decrease. So that would be a full description of what's happening here. Now, we're going to have a look at this data from another angle. So we've got a table here, and our question is, which state has the highest number of mesothelioma cases? And also, in which state do more people per 100,000 get mesothelioma? So there's quite a bit of data to wade your way through there. So I'll just give you 30 seconds to have a look. Okay, let's dive in and see how we've gone. So, which state has the highest? New South Wales. Now, does that make sense? Probably, because we've got a lot of people. And which state do more people per 100,000 get mesothelioma? And of course, that's Western Australia, because their rate is 4.9 people per 100,000. So if you got that, fantastic job. Now, the next thing we're going to have a look at is melanoma. So remembering that mesothelioma is environmental, whereas melanoma is environmental exposure to UV as opposed to asbestos. So melanoma is a type of skin cancer that develops in skin cells called melanocytes and usually occurs on parts of the body that have been overexposed to the sun. Rare melanomas can occur inside the eye or parts of the body that have never been exposed to the sun, such as the palms of the hands or the soles of the feet or under the nails. So what we've got here is a 5-year prevalence. Now, remembering that prevalence is how many people have it or within a particular time period, as opposed to being diagnosed with it, which would be incidence. So here we're identifying the prevalence of melanoma for males and for females in 2014. So I'll just give you a second to look at that. Now, again, when you're answering these sorts of questions, I would literally get my pen and draw a line over to the edge. So what we can see here is that we've got 32,000 males and about 23,000 females. Here again, we can see melanoma incidence and mortality. So here we've got the incidence, so how many diagnosed cases in particular years, and a projection. And we've got how many people are dying from this disease. So what is the predicted incidence and mortality for melanoma in the year 2040? So we can read those numbers off and provide a possible explanation for your prediction. So one of the things that I've noticed in biology, not only do they want you to take your information that you have and be able to apply your understanding to a new situation, you need to be able to come up with reasonable predictions. So we may never have learnt in the syllabus why we think that's happening, but if you use your logical mind and say, well, what can we come up with? Let's have a look here. So we can see that our incidence is 475,000 people. Our mortality is 100,000 people, and our explanation, now, as long as it is reasonable, you would get a mark. So many more people are getting melanoma as the damage from sun exposure has already occurred, but death rates are only increasing slowly as treatments are getting better. So I'm sure you've all received the sun-safe messages. I, on the other hand, used to lie with coconut oil in the sun so I could be brown because it missed me. So I'll have to look out for melanomas in the future. Now, we've got another type of disease here, which is malnutrition. So we're going to jump forward and we're going to have a look at page 33. So malnutrition refers to deficiencies, excesses, or imbalances in a person's intake of energy or nutrients. The term malnutrition addresses 3 broad groups of conditions. So we can have undernutrition, which refers to like wasting or stunting. And so wasting is low weight for height, whereas stunting is low height for age, and underweight, low weight for age. We can have micronutrient malnutrition. So you might have a vitamin A deficiency or a vitamin D deficiency. And of course, there's overweight, obesity, and diet-related non-communicable, which just means non-infectious diseases, such as heart disease, stroke, diabetes, and some cancers. So what we're going to do here is we're going to have a look at this particular graph and say, identify the trend for stunting in children under 5 globally from 2000 to 2020. So there's a lot of information in there. So let's have a look at it. So identify the trend for stunting. So if we have a look down here, the yellow is stunting, and they want to see what the trend is between 2000 and 2020. And as we can see here, it should be coming down. So the percentage of children with stunting has decreased from 33% to 22% over 20 years. Now, the next one, this table, again, quite confronting, lots of data in there. Let's identify the total number of children under 5 affected by severe wasting in Africa in 2020. And when we look at this, we can see here, we're looking at wasting and severe wasting. So we've got severe wasting here, and there was a whole bunch of information, but I've just given you a few here. So if we go across to Africa, we can see that our answer there should be 3 million children in Africa have severe wasting in 2020. Now here, what proportion of children under 5 are affected by stunting in 2020 who live in Asia? So again, let's have a look and see if we can wade our way through this. So we're looking for children under 5 who are affected by stunting. So we're looking, and they live in Asia. So I think yellow is still stunting, hopefully. Here we go. And 53 or more than half the children affected by stunting live in Asia in 2020. Now, this question is a really important one, I think, because it really shows an ability to think critically. So the question here, what we can see is we've got a prevalence of vitamin A deficiency in children. And so what we can see is that the dark red is there's, up to almost 100% of the children have a vitamin A deficiency. We've got all the way down to the yellow, which is not much at all. And then we've also got places with no data. Now, this question asks us to compare the prevalence of vitamin A deficiency in Central Africa and Australia based on the data provided. So if we look at Central Africa, there's a lot of vitamin A deficiency. And when we look at Australia, what do we see? No data. Now, your inclination might be just to say, well, there's less in Australia, but what's really important is to say that there is actually no data for Australia, so a comparison is not possible. Now, if you want, you could then say Central Africa has areas where up to 100% of the children under 5 are affected. And I do think, given that the socioeconomic circumstances in Central Africa are lower than they are in Australia, we would expect to have less here. But if we're going by the data on the page, then we can't comment on that because we don't see it. Now, moving on to my favourite part, I'm just going to check how we're going for time. Oh, it's 2:53 already. Okay, so I'm going to whiz you through this one. So this is my favourite part because it gives me an opportunity to tell all of you not to vape. So remember that cigarettes used to be considered really healthy. So there were actually advertisements saying more doctors smoke Camels than any other cigarette. And they were really saying, oh, it tastes really good and, it really helps asthma and all of these things. And now we're in a situation where cigarettes are so unhealthy, they'll give you cancer. So my question to you is, how did we get from here to there? So when we look at epidemiology, we're looking at, if we literally translate it, it's the study of people. So we use the term epidemiology to mean the study of diseases or other health-related events in populations. So here's what I want you to think about. We know that smoking causes cancer because we have a whole lot of data. Epidemiology allows us to answer those questions. Now, I'm going to ask you, does vaping cause cancer? I want you to ponder on that for a second. So here's where we are now. We've got vape companies that go, oh, look, kids, these are fun and tasty. They're just like lollies. I honestly think they're doing the same thing that they do with alcohol. They get your vodka cruisers, your Bacardi breezes, all of these things, and they put pretty colors and pretty flavors in them, and they try and make them appeal to you because teenagers are like, oh, fantastic. It's bright and shiny and it's fun and it's new, because your risk-taking centers have not fully developed yet. And that's a good thing because it allows you to take some positive risks. But I'm going to say that I think these companies are playing you all for fools, because I think that they don't believe that you will understand the difference between vapes and cigarettes, because you'll get people saying, oh, it's just a vape, it's really healthy. So The CDC has declared an outbreak of the e-cigarette or vaping product use associated lung injury. So they call that EVALI is the acronym. And so essentially what they're seeing is massive problems with people who are vaping a lot. But is there a heap of data? And the answer is not yet. But what I would like to say to you is some chemicals present in e-cigarette aerosols are capable of causing DNA damage and mutagenesis, which you all know about, which supports the biological plausibility that long-term exposure could increase cancer risk. Now, I also wanted to say that I've noticed probably in the last 3 or 4 months, the Lung Foundation are now putting out public health campaigns saying that we possibly shouldn't vape. Now, case studies have suggested that e-cigarettes interfere with or delay wound healing. So I actually know someone who went in for surgery recently, and the first question the doctor said is, do you smoke or vape? Because you need to stop before we do this surgery because your wounds will not heal nearly as well. Now, also the evidence for a strong positive relationship between the use of e-cigarettes and later cigarette smoking amongst youth continues to accumulate. So what they're saying is the data is coming in, and I do think that we are going to land up with vapes just the same with cigarettes. Oh, it's not bad for you. Oh, yes, it is. It causes cancer. So this is the conclusion from the CSIRO, which is one of our peak bodies for science, and the evidence available suggests that the regular use of e-cigarettes is likely to have adverse health consequences. There is a lack of clarity about the magnitude or the size of these adverse health effects and the quantity of e-cigarette use required to trigger adverse health effects. So just like regular cigarettes, if you smoke a couple of cigarettes, that is not going to kill you, but continued use of vapes, I suspect, is going to be really harmful. And I do think that as biologists, it's really important that you critically consider the evidence available. So when we look at epidemiology, we've got a few different aims. We want to describe disease patterns, we want to identify causes of disease, and we want to provide that data that's essential for management of evaluation and planning. Now, I'm just looking, we've only got a few minutes left, so I want to take you to probably, this is something that I always found tricky when I was first learning it, is the types of epidemiological studies. So basically we've got these observational look, don't touch studies where we can see a case report. Oh gosh, look at this interesting thing. We noticed this person got this disease, or there's a case series where we've noticed a couple of people have got these diseases. Okay, so these are descriptive studies, just having a look at what are the circumstances. We've also got these analytical studies where we're looking at a cross-section of people at a moment in time. So I'm just going to flick over. So for these cross-sectional studies, we might look at, for example, people who have lung cancer and people who don't have lung cancer and see what's different in their lives. And of course, smoking will be one of those things. Now, case-controlled studies, we're going to have a look at disease status. So, sorry, I beg your pardon. So cohort studies, we're going to look at the risk factors and follow over time. So these are all types of analytical studies, whereas when we move on to the other side, we're looking at these experimental or interventional studies. So this is where they might actually give you a tablet or a placebo. If you're doing a double blind, we're giving you a tablet, but it's actually a sugar tablet, and seeing what's happening there. So what I would suggest to you, since we're just about at time, is there's a whole bunch of information in here. There's some questions, there's some answers. Your teachers have access to not only the workbooks, but they will also have access to these PowerPoints. And it's something that you can have a look and work through as part of your HSC preparation. So my apologies that my timing was a little bit off today, but there's a whole bunch of stuff in there for you. I'm not sure if anyone has any queries or questions before we finish up today, but I would like to say thank you very much for coming along to this presentation.
Thank you, Halima. Thank you everyone for attending. Look, there is so much stuff to go through, and Halima, can I be honest, last year was worse. You did much better with your timing this year. Last year it was like, there's so much to get through. So yeah, no, fantastic presentation. Thank you so much. I hope you all got something out of today. There are so many tricks and tips in there for your upcoming trials, HSCs. Good luck to everyone. Thank you, all the teachers, for organising it. Thank you to our wonderful presenters for the amazing work that they do and the fabulous resources that they're sharing. We wouldn't be able to do it without them. So thank you again, Halima.
My pleasure, and good luck, everyone.